Hi there, welcome to this video. This is the second one in the series of videos going through these past exam questions. So the next section of questions is linked to the PAG on reducing sugars and non-reducing sugars. So question 11, an unknown solution of a single sugar was tested. So it means we're only testing for one sugar, not a mixture of sugars. The results were recorded in table 9.1. So when they did the Benedict's test for reducing sugars, it turned blue. And when they did the Benedict's test for non-reducing sugars, it turned brick red. So for the first thing that you need to know is the result colours. Okay, so we've got this colour scale. So it goes from red if there's a high concentration of it, to orange if there's slightly lower concentration, to yellow, slightly lower concentration again, green if there's a low concentration, and it stays blue. That basically means there's been no colour change and there is no concentration. So if you look at this one has stayed blue, so that tells us that there's no reducing sugar present. Okay, whereas brick red is right at the top of the scale, so that tells us there's a really high concentration of reducing sugar. Okay, so we need to know that colour scale. So identify the unknown sugar. So let's have a look at what these are first of all. So Benedict's test for reducing sugars, you straight away just look at the colour scale and it tells you that it's blue. So that tells us that it's not a reducing sugar. Okay, it's not a reducing sugar. Now, hopefully, you would know that glucose is a reducing sugar, so it cannot be glucose. Now, glucose is, as we know, a monosaccharide. Hopefully, you can remember your disaccharides. So remember the ones you need to know: maltose, sucrose, lactose. So maltose is glucose and glucose, sucrose is glucose and fructose, lactose is glucose and galactose. Okay, so sucrose is what we call a disaccharide. Lactose is a disaccharide, and fructose is a monosaccharide. Okay, now monosaccharides tend to be reducing sugars. So the fact that we found that it's not a reducing sugar, it stayed blue with the reducing sugar test, tells us that it's not going to be fructose either. So then we're left with two disaccharides, lactose and sucrose. And in this test over here, this was the test for non-reducing sugars. So you might remember that when you did a test for non-reducing sugars, what you do is you do the test for reducing sugar, you get a negative result, which means it stays blue. And then what you do is you add some hydrochloric acid. What the hydrochloric acid does is it breaks a disaccharide, like that, into two monosaccharides, okay, because it breaks them apart. You then have to neutralize it. Okay, and then you do the reducing sugar test again and you look at the colour scale. So what's happened in this case is that we've started with a disaccharide that wasn't a reducing sugar. When we did the non-reducing sugar test, which means we add hydrochloric acid to break it up into monosaccharides, then it was a monosaccharide that was able to change the colour to reduce the Benedict's reagent. So we would need to know. It's hard because you do have to remember it really. The correct answer is C, because sucrose, we should know, is a disaccharide that's a non-reducing sugar, because when it's broken down it produces glucose and fructose, and those are both reducing sugars, and therefore the colour then changes. But ultimately you need to remember that monosaccharides are reducing sugars, that sucrose is a non-reducing sugar, and lactose is not. Okay, but it is tricky to remember. The main thing to remember from this particular question is the colour scale for your results, what they tell you, and the method of how you test for reducing sugar and non-reducing sugar. Okay, let's go down to the next one. Some organisms use a disaccharide called trehalose as a respiratory substrate. So we've talked before about how they're going to use molecules that you haven't learnt about to see if you can apply what you have learnt to a new context. So they use it as a respiratory substrate. That means that they use it for respiration. Okay, so they use it in respiration. They break it down to try and produce ATP to release energy. Trehalose has a similar structure and very similar chemical properties to sucrose. Okay, so we've just learned how we could test for sucrose to get a positive test, and now it's just asking you for that method. So we know that sucrose is a non-reducing sugar. So in order to test for a non-reducing sugar, first of all, you test for a reducing sugar and you get a negative result. So first of all, you do the Benedict's test, which is when you add Benedict's and heat. And you should get a negative result, which means that it just stays blue. And then you do your second stage, so you're going on to testing for a non-reducing sugar. So then you heat it with hydrochloric acid and neutralize. Then you would retest with Benedict's. So add the Benedict's and heat again. And this time you should get a colour change. Now, if there was a high concentration, it would go brick red. But remember, it's still a positive result if it goes orange, yellow, or green. They're all positive results. They're just different concentrations of it. Okay, so make sure you know how to describe that method. On to the next question, 13. This was a tricky one. So lactose is a reducing sugar. So this is interesting because lactose is a disaccharide, okay, but it's actually a reducing sugar. Now, usually, most of the time, we learn that the reducing sugars are the monosaccharides. Now, if you wanted to kind of clarify why it couldn't be the correct answer up here, okay, is because in the first test it stayed blue. Now, lactose is a reducing sugar, so when you've done the first test for reducing sugars, lactose would have tested positive, and that's why it couldn't be lactose. Even though it is a disaccharide, it's a reducing sugar disaccharide, which is really unusual. So it's a disaccharide that is a reducing sugar. Okay, I don't know why there's an S on there, sorry. Which is unusual, but it tells us that that's the case. We know lactose is a disaccharide because we know that maltose, sucrose, and lactose are our disaccharides. We need to learn those. Benedict's reagent can be used to detect the presence of lactose in a solution. A colorimeter can be used to measure the concentration of lactose. The colorimeter first needs to be calibrated. Describe how a method that uses Benedict's reagent and a colorimeter can be calibrated to measure the concentration of lactose in an unknown sample. Now, any experiment you do where you get a colour change, you can quantify it using a colorimeter. Now, results come into two categories. Some experiments give you what we call quantitative results, which means that they give you results that are numbers, quantities. Other experiments give you results that are what we call qualitative, and that means that there's no numbers. So, for example, a qualitative result could be a colour. Okay, so the way we did these experiments was a qualitative investigation. Now, sometimes you could say it's semi-quantitative because you've got the colour scale where you've got the high concentration is red, lower concentrations orange, lower concentrations yellow, etc. So there is an argument that it's semi-quantitative, but it's not quantitative. We're not getting exact values. We don't know exact values. We haven't got any numbers. So The first thing is understanding obviously how you would test for reducing sugars, but really it's more about the calibration and the colorimeter. Now, hopefully you've used a colorimeter in a different experiment and you can describe how you would use it. So when you use a colorimeter, first of all, you need to zero the colorimeter. And that means using usually water. Okay, so you can use water, and it's just setting what you want zero to be. Now, if you put one of those little square cuvettes, they're called, those little square containers in the colorimeter with water in. Now, we set that to zero, which means that we're saying that when we put water in, there's 0% absorbance. Now, reality is that little plastic container containing water would absorb some of the light that you shine through it. But we're just setting it, we're calibrating it to say that that's zero, just so that we've got something to compare to. Okay, calibrating means you're deciding what to compare it to, you're deciding what zero is. So what we do is we put water in the little cuvette, we put it in the colorimeter shines the light through it, and we say whatever that reading is, let's call that zero. So that's called calibrating it. Okay, so zero the colorimeter using water. You could call the water a blank. That's another word for it when you just use like the equivalent of no substance that you're trying to measure. Whenever you talk about colorimeters, we normally try to specify which filter. Most of the time we use the red filter. If you're in doubt, say you use the red filter on the colorimeter, unless you happen to know otherwise. Now, the way that we can quantify this from an unknown sample is to first of all test on the colorimeter a sample that we know. So what we would do first of all is we're trying to find out the concentration of lactose. So the question is to measure the concentration of lactose. So what we need to do is we need to, first of all, put some concentrations of lactose that we know. So if we got some known concentrations of lactose, and we got concentrations of, let's say, 0%, 20%. We always try and do 5. That's good practice. 60%, 80%. And then with those concentrations of lactose, you perform the Benedict's test. So you add the Benedict's solution, you heat it up. Then what you're going to end up is 5 samples of different colours. Now, the one that's 80% lactose has got the most reducing sugar in it. So that's probably going to go your brick red. Okay, this one is probably going to be a lighter red. And this one might be orange, and this one might be a light orange, and this one might be yellow, for example. And it might be that they've all got quite a high concentration. They all just look red to the eye, but maybe the colorimeter can tell the difference between how dark the red is. So then what you do is if you put those into the colorimeter and you test those on the colorimeter, and what you do is you then draw those onto a graph, colorimeter, and draw on a graph. And you're going to end up with a graph that's got a series of points. If you've got your concentration there, the darker red is going to the darker red one up here with the highest concentration is going to absorb the most. So you're going to get everything up here. The light red one is going to absorb less. The orange one's going to absorb less. Light orange is going to absorb less. Or yellow is going to absorb the least. So this is your absorbance reading, which is in percentage from the colorimeter. And we should call this graph here a calibration curve. Okay, so then what you can do is you can test your unknown sample. So then you're going to get your get your unknown sample, so the lactose solution that we don't know how concentrated it is. And with that one, we can add Benedict's, heat it. It's going to change colour. We don't know what colour it's going to go, but depending on what colour it goes, what we can do is put it in the colorimeter, get an absorbance reading, and look this up on our calibration curve. Okay, so for example, if you did your if you got an absorbance reading that was something like this, then you can draw across, draw down, and you can read off what the concentration was, 0.58 or something, whatever it is. Yeah. So you then, whatever your reading is, you go across till you hit your line, go down, and you can read what the thing was, what the concentration was. Now, if you draw a line of best fit on here, which you would do, if you've got a line of best fit on there, excuse my freehand drawing, when you got your reading, even if you got a reading that wasn't on one of your X's, you just, whenever you hit the line, you go down and then you can read the concentration off there. And even if it's not one of the concentrations you tested, you can still read it off on the scale as long as you've plotted it using a scale. So you can then predict that this is the concentration of my unknown sample. Okay, that is a really tricky question, that one, but it's getting used to this idea of how we make calibration curves to test unknown samples, and that can apply to any experiment where you're looking at colour change. Okay, so it's worth knowing. The other thing that's worth knowing from that question is what's the difference between quantitative results and qualitative results.